Okay, Biology 115, Part 2, Proteins, Formation of Peptide Bonds. So first off, we're going to define residue. We're going to identify the N and C terminal residues in a peptide, explain the structure of the peptide bond, forgive me, and I justify why, although hydrolysis reaction is thermodynamically favored in an aqueous environment, peptides are typically quite stable. And you're going to differentiate the terms peptide, polypeptide, and protein by the end of this lecture. Okay, so jumping right on in, you should recognize the structures drawn down here. These are amino acids. And as you have learned previously, amino acids can combine to form proteins. This reaction is called a condensation reaction or hydrolysis reaction because when we line up the amino of one amino acid and the acid of another amino acid, we have our water leaving group minus H2O, and this will allow the formation of a new bond between the carbonyl carbon and the amino nitrogen, forming, let me draw that first, C, N, now back to the other color, the boring color, H, C, C, H, nothing else will change, but this right here is our peptide bond shown in purple, the line shown in purple. Goodness. Okay, C, N, H3N plus, and the side chains do not change. Okay, so here's the formation of our peptide bond done via hydrolysis. In terms of entropy, we're taking two things and creating one thing. So this is more disorder and this is more ordered. So this is less disordered. That's not very good until you consider that an additional molecule was made, H2O. And then this can go out and join the chaos in the rest of the water. That's approximately 55 molar in every cell that we're concerned about. So in terms of that 2 to 2, it's 2 molecules to 2 molecules. At least it's the same level of entropy. But in reality it's increasing because this has much more entropy than either one of these two, especially in an aqueous environment. However, thermodynamically speaking, this reverse reaction would be extremely, extremely favorable. And if we look at the delta G of it, and the delta G naught prime, it's in fact quite favorable. However, the delta G does not take into effect temperature. This is favored at a higher temperature. And unless this reverse reaction is catalyzed by an enzyme, it pretty much never, ever occurs. The peptide bond is quite stable. So I'm going to draw a smiley face. It is quite stable. Boom. So happy, right? So once it's formed, unless something is acting upon it, unless an enzyme is acting upon it, a protease, for example, this peptide bond is there to stay. Okay, a couple more definitions. So, residue. I've been saying residue. Some of you might be confused. What a residue is what's left over, like after the water evaporates, right? Like the residue of your coffee will turn your coffee mug black. So after the water is gone, what's left is the residues. So the residue is an amino acid joined with a peptide bond. With peptide bond. Okay, so these are the amino acids in a protein, are called residues because they've undergone this dehydration reaction, peptide bond formation. I'm sure there's several other names for this, I just can't think of any at the moment. So that's what a residue is. So attach that to your vocabulary, start using it frequently. So next up, we're going to label the N and C terminus residues. Okay, so the N-Terminus N-Terminus refers to the amino terminus. So that's the one with the free amino group. This is typically designated as blue. The C-terminus is the one with the carboxylate group, typically designated as red. So this is the C-terminus of this short peptide. And it's really just what's exposed. So you can see this can't be the C-terminus because this carbon of the carboxylate group has been used to form the peptide bond. So N-terminus here, C-terminus here. So we typically orient all of our proteins, and this is just the convention, we always list the residues in the primary structure from N to C terminus. So I remember that the N-terminus is typically blue and the C-terminus is typically red, because red is stop, right? So we use stop at the C-terminus. Okay, and then the last thing we're going to talk about is differentiating peptides, polypeptides, and proteins. And this is really based on the number of peptide bonds. So peptide is true for any short sequence, this is the shortest, short sequence of amino acids joined together. It's typically less than 15 residues. This is also sometimes called an oligopeptide. So just a few. Because if you remember, oligo means few. So just a few peptides joined together is called a peptide. A polypeptide has, and this is all really approximate, it's not very scientific, but this is what we've got. It's about 15 to 50 amino acids in length. So peptides are the shortest, then come the polypeptides, and then come the proteins. Proteins are typically more than 50 amino acids in length. And a lot of people feel that just the proteins have this catalysis function, that they're able to do chemistry, but that's not true at all. Peptides, polypeptides, and proteins can all have catalytic functions based on their primary sequence. So depending on what residues are there, what amino acids have been joined together, that will give the proteins, peptides, polypeptides, different properties that could function in catalysis or structure.